Now we would like to start the physical quality module, which is essential as determined by our specific standards. It is essential for issuing international certificates, and it's one of the certificates that requires training the analysts the most, and it also requires a lot of time. We would like to welcome Fabian Mankowski. He's been working at INASE Lab since 2014, and he will be making a presentation. During his presentation, we will be receiving your questions from the chat, and we will ask him the questions at the end of his presentation. Thank you, Vanessa. Well, good morning, afternoon, or evening. I would like to tell you about the requirements as set by the specific standards. What's a standard? I looked up a quick definition on Google. Google says that standards are specifications on how a task or role needs to be developed, and they are based on agreements between one or more entities or a group of people. Based on this definition, what are specific standards? According to Decree 438 of 2014, which governs seed law number 16811, as Daniel told you before, our foundational law. The specific standard is a document proposed by the board of the National Seed Institute, approved explicitly or tacitly by the Ministry of Fisheries and Livestock, which establishes the characteristics and requirements for seeds and associated processes, especially regarding the production, trade, etc. These are minimum rules that define the requirements that seeds and related processes need to comply with. This is a tool agreed on by the stakeholders that participate in the process. We are talking about breeders, seed companies, seed multipliers, seed consumers, and related institutions such as the Research Institute or the School of Agronomy. These standards are defined by a multidisciplinary and interinstitutional technical committee. It uses strategic plans set by the Ministry of Livestock and on international agreements as well, and they define the next steps to follow. First of all, they are approved by the board of INASE, and then they are approved by the Ministry of Agriculture, Fisheries and Livestock. What type of information is included in the specific standards? Common scientific name of the species, date of the latest update, field parameters, laboratory parameters, and other observations. In this talk, I will focus mainly on laboratory parameters. These standards ensure quality. What type of quality? Genetic quality that has to do with the behavior of the seed, also physical quality that has to do with the physical integrity and composition of a seed lot. This includes, for instance, a pure set, which is the seed that we are interested in, also an impurities section that might include other seeds that correspond to cultures or weeds, and other inert materials such as vegetable remains, earth particles, or maybe pieces of the seed we're interested in, but that do not comply with the pure seed definition as provided by ISTA. There is also physiological quality, which is the viability that a pure seed lot has to germinate. This seed pure is germinated and under favorable conditions, as determined by ISTA in its rules. And after this, we're going to evaluate this germination power. And then we have this health quality, which means that they're harmless and do not transfer any pathogens. This is essential for reproduction. Classes and categories. The formal seed system divides seed lots into different classes and categories, and the standards determine the requirements for each one of them. Standards, classes, and categories, and also the ones that correspond to the systems. We have two categories, pre-basic and basic. They have the same requirements in the standards, so we will address them jointly here. Pre-basic seed through multiplication will give rise to the basic seed. Then this will lead to certified 1 seed, then certified 2, and then commercial A. And we have the category then commercial B. Why is it different? Because it doesn't have a generational control. It may come from a certified seed or from a seed that falls outside the certification programs. Here we can see that the first categories fall within the certification scheme, and the second, the last two ones fall outside the certification scheme. How are these categories different? Certified categories are controlled. Actually, the production process is fully controlled by INASE directly through experts or indirectly through INASE, as it accredits companies that need to comply with certain processes and procedures. And through them, INASE accredits the companies and the process is controlled. And we have the commercial categories. And the companies and the company experts are responsible for these seeds, and INASE participates once these seeds are sold in the market. Where can we find the standards? On INASE's website, we can find them where it says Normativa in Spanish, regulation, and there we have the production and trade standards. If we click here, we can see the different standards that are classified into agricultural crops, forage grasses, and also vegetation propagation material. For instance, if we click on the rye standard or a specific standard, we can then, on the barley standard, sorry, we can then download the information. Here we can see how the different species are grouped. And now we have the standard, and this is what is important about this topic. Which different parts can we see in a standard? First of all, the heading.
This is important because it gives us the date of the standard and the last update. It is important to see that we are following the last standard approved. That's important because of the market requirements, and so we have to really see that it is updated. Then we have the field parameters that is not of interest for our lecture now, and then the lab parameters. Here we have the maximum weight of the lot. Here we can see it's determined by the ISTA rules and the minimum weight of the sample to be sent to the lab, to submit to the lab, and then the minimum weight for the work samples for the physical purity or to determine other seeds in number. Then we will see the different categories that will be differentiated. We have pre-basic and basic that are together, and then certified 1 and certified 2. As you can see, the most basic categories, they have more strict requirements or standards. And then for each of them, we can have the percentage of pure seed, the inert matter, the amount of other seeds that the lot can have, and so on. So here we will put 2 examples. We will have with this example of alfalfa. These are the examples, the results of our test. and we will compare it with the standard to see if it complies or not. Here we have alfalfa's standard, the latest updated version. What we have to do that many times analysts are not doing, before starting the analysis or the test, we have to check the standard, see what requirements are placed in the standards, what we have to focus on and what is not so important, because each species has different characteristics. And as analysts, we have to know, for instance, here for alfalfa, we have to know that due to a national law, we have to make an extra analysis that is seed with a higher weight than the usual weight for analysis in other seeds. Here you can see that it has to be of 500 grams in this case. This is something that nobody will tell you. The standard makes it clear, but you have to know that in advance so that you don't make the mistake of doing first the testing, and then when you go and compare it with the standard, you see that the part of your test were missing, or there was weed that you had to look for and you didn't, and so on. So let's go to the important stuff. So we have to first see that this is a species we are looking for and we are trying to test. Then we have to go step by step. First, it tells us that this is a C1 category. So let's go to the standard and see where it refers to certified. And we see the standard. We have to see that it's different for C1 than C2. So for pure seed, the minimum acceptable is 95%. We see our results, and we see that our results is 99.7%. So here we are complying. Then we check inert matter. Maximum accepted is 5%. We check our results and we see 0.2%. So that's fine. And then we continue with other seeds. The maximum is 0.6%. In our data, we have 0.1%. So we can say that it complies. So let's continue. We have to analyze, to go to the test of, or to determine other seeds in number. Here, there are some points that we have to pay attention. It states which are the weeds that are accepted for alfalfa. This is important because for each species, they are different. It's a big difference for the ones that have zero tolerance weed. That is, for many, they are the same. But for rice, we add one, as Vanessa was pointing out before. So in this analysis, we find 6 seeds of Maxus, 3 of Rumex XP, and 8 of Polygonum avicular. If we check the tolerated seeds, we see that we find AMI and Rumex. So we can say in total that in our analysis, we have 9 seeds that are tolerated for this category, but the standard says that we can have for C1 up to 15. So that means that we are complying with this item. We are meeting this requirement. So then we have the possibility of Cuscuta, 500 grams. That is something that it doesn't have. It is a forbidden one, so we are doing fine. And then, well, here we have the zero tolerance weed that I was referring. And for germination, it has to be treated with a minimum of 80%. And for this lot, our test shows is 92% germination. So we can say that our results, according to the standard, they are meeting all the requirements. So we are according to the standard. Let's go to another example here. We would like to add some details that should make it a little more complex. Here we have the latest standard for this rescue grass. We look for certified one, that's the category, and we will go item by item. It says that the minimum is 96% of pure seed. Here we have 99.4%, so that's okay. We meet the requirement. Inert matter maximum 4%, and in our results we have 0.3%. So it also meets this requirement. Now we go to other seeds. We are in physical purity. Here it allows 0.5%, and in our results we have 0.3% of other seeds. So here we meet the requirements. So we can move now to the next analysis, that is to determine other seeds in number. Here in the example, I put the seeds that were found. It's important to detail this in the analysis, but in the physical purity is not so important unless it's a forbidden weed. So here we have the list of the weeds that are tolerated, and we go and compare. We see our results, and of this list compared to our result, we see that the only one that is tolerated is Cirsium vulgare, only one. And the standard states that we can have up to 10 of the tolerated weed. So here we meet the requirements. Then we have other weeds from other cultures, and we have to see which others are of interest for this species. One is the different types of oat, Lolium, hybridum, and we compare if we find any of these seeds in our analysis, and we find the two types of this oat and raygrass. So we check, and we have 9 seeds, and the standard informs that we have up to 10. So here we are meeting the requirement because we don't go above this value. If we continue going down in the standards, the number of seeds that we have to go to item 6, it's another type of oat. And we go to our analysis, and we found 4 seeds of this fatwa oat, and so we have 4 seeds of these ones in the standard. So if we compare it with the standard, the maximum is 3. So here in this item we are not meeting the standard, and so that our results are according to the standard, we have to comply with all the requirements. And so we can say that for this category, we are not in the standard. Here we check germination. That was the last one that we had to check. This says the minimum to trade is 80% germination, and our result was 98%. So that's one that we meet. But as there was one of the requirements that we didn't meet, we have to say that this is not meeting the standard. So here I made a checklist to what to look when we compare our results with the standard. We have to check the last update of the standard if it's in force. Then we have to see if it complies with the minimum percentage of pure seed, that it doesn't go above the maximum percentage of inert matter, that it doesn't go above the percentage of other seeds, that it doesn't have a 0 tolerance weed, that it doesn't go above the maximum of tolerated weed, that it doesn't go above the maximum number of other seeds, other cultures, and that it complies with the minimum germination requirement. And just to close, I would like to share a sentence that I like, they like very much. I was told and I forgot, I saw it and I understood, I did it and I learned. This is a good summary of what the analyst is doing, and the analyst's life is not only to listen and to just do it. We have to be proactive. We have to learn on our own, and then we put our hands on and we go and have the practice and move forward. Thank you very much. So this was very good. Thank you very much, Ignacio. Your contact is shared. And the standards is, the analysis according to the standards is a huge responsibility. And we have to use this information in the right way. So there were some questions that I will ask you. If I test a sample C1 and it comes out of the standard and it complies with C2, can I change the category of the seed? Who is making that decision? Well, it can always be put to a lower category for C1 to C2. The decision is made by the company. It can happen that the company doesn't want to lower the category and they would like to reorganize to try to meet the standard, but that's a company's decision. That can happen often, so it's a good question. Second question, why are there specific standards for species that don't have tolerated weed? Well, that depends on each species, because on the specific characteristics of the species and the production. Remember the sorghum standard that doesn't have tolerated weed. In the physical purity, in the percentages, it's more demanding. And requirements are more strict, and it depends on the weeds that are a huge problem and that cannot be controlled in former stages. So thank you. The next question we have is if the sample set is a lower weight than the one set forth in the specific standard. Can we test it anyway? That's a very good question. That happens quite often, that the sample doesn't comply with the minimum weight. And in that case, we cannot carry out the testing. We have to require next sampling, not to complete the weight that is missing, but a new sample with the minimum weight of the sample. And that weight you have to comply with that weight. If not, you have to do a new sample. Thank you very much, Fabian, for your presentation. It was very clear. We keep your contact data. We invite you to continue here to enjoy this day with us. It's a very important topic that has a lot to do with our country, as we have so many people from other countries. If you are thinking about exporting to Uruguay, you have to know that there are standards for the different species. And this is also important for our national technicians, that they have to be very clear about how to deploy the standards. I would like to introduce you to Ana Tardagila. She's an agronomist. She studied at the University of the Republic, and she is responsible for the physical physiological lab at the INASE, and she is going to tell us what every analyst must know. Good day, good morning, everybody. I hope you're enjoying this meeting today. We are going to handle some points that we believe that every analyst must know, and many times they are not referred to because the priority in the training of analysts is focused on the different techniques they have to follow and learn. So the first one, why do we have seed analysts? The seed-related activity is considered a critical step in the agricultural production and productivity. Regarding the importance of this activity, there are rules that govern this activity and provide minimum quality requirements. And the last purpose of this is to assure farmers a proper access to quality seeds. So as there are specific and minimum quality requirements in Uruguay that we find in the documents that were referred by Fabian in the former lecture, we need to check that the seeds coming to the farmers, they comply at least with the minimal requirements established in the requirements. So the seed testing labs, they are the centers where this quality is controlled. So without that work of the analysts, seed analysts, the regulations or the rules for the seed industry couldn't be applied. What is the responsibility of the analyst? The main responsibility of the analyst is to be very strict when carrying out the tests so that they avoid two mistakes. One is to reject a good lot that complies at least with the minimum requirements of the standards, with the problems that it can bring to the company that is breeding these seeds. There is a lot of inconvenience, like the production will be delayed and other lots will suffer. And the other mistake is to accept a lot that does not comply with the quality standards. And so such a lot would be used and for production with a bad quality. They will bring extra additional costs for the production to the farmers. So what does it mean to be strict when carrying out the tests? It doesn't mean to be so strict or mean with when we do the tests. It's just to follow very strictly the standards published by ISTA, as mentioned by Ignacio in his presentation. If we follow this methodology, this standardized methodology, we will be sure that our results will be exact and trustable, reliable. They could be reproduced and compared. That is, the results that are obtained in one lab with one sample, they could be reproduced with some accepted tolerance with another sample in another lab. And also that the results of the tests are understandable. That means if all analysts, they apply exactly the same criteria when they carry out the tests, the results that are informed, they have the same meaning for everybody. So they are understandable everywhere. So to follow strictly the procedures, the standardized procedures, that doesn't assure that we will have exact or reliable results if we don't give the importance to the sample for working. The sample to be analyzed, it must be a real representation of the lot. So as in the lab, when we mix and divide the seeds, they have to be done exactly following the procedures. If the sample that is delivered to the analyst wasn't not obtained in the proper way, then all the efforts that the analysts can make when carrying out the tests, they won't be good, because the results, they will be on a sample that is not a sample that is really showing the composition or the quality of the lot. So finally, probably the decisions will be wrong. The decisions on the lot will be wrong. So what is a seed? If we ask a botanist about the meaning of a seed, they can come with one of the three definitions or all the three. It will be the result of the transformation of an ovule that is that a seed is composed by the embryo that will be originating the new plant, the nutrients that can be in another tissue, an independent tissue, the endosperm generally, or they can be incorporated in the embryo once the seed is ripe, and they will be used in the first stages of the development once germination starts. And embryo and nutrients, they are covered by testa, which is the cover, the coat of the seed. So this can be also told that is the structure that is in charge of spreading and perpetuating the species. This function in the plant is not carried out exclusively by a unit that corresponds exactly to this botanical idea of the term. I don't know if the popular or agronomic idea of seed is the one we use usually, that we use it to refer to botanic seed or fruits that have one seed or parts of a fruit, or even similar to fruit things, structures that include parts, some other parts besides the fruit. So the seed analyst has a unit that is a spreading unit that is very, it has a variety that doesn't necessarily follow a pattern that represents that unit or the parts.
This actually faces analysts with the main challenge within his or her activities, the species a seed belongs to that they cannot detect visually. 90% of the plants in the world nowadays belong to the angiosperms group. Most cultivated species belong to these species and also to the weeds. This group, angiosperms, includes around 300,000 species, which are distributed into approximately 400 families. Of these families, the Asteraceae family is the one that has the most, the highest number of species, 24,000, and it is widely spread in the world, everywhere except the Antarctic. The fourth family regarding the number of species is the grasses family. It is the most important one from an economic perspective, and it has representatives throughout the world, and it even has some species in the Antarctic. 300,000 species is a lot. When an analyst finds a seed they can't identify, we can say that they have 300,000 possibilities. If the analyst can recognize at least the family the seed belongs to, in the worst case scenario, they would be facing 24,000 species, which is the number of species included in the Asteraceae family, the one with the highest number of species. Therefore, it is beneficial for the analyst to be able to recognize, to detect a seeds family, because it drastically reduces the number of possible species. It increases the possibility of identifying the species accurately, or to report the genus. Or maybe they can report which family the unknown seed belongs to. And of course, as the analyst fine-tunes the search according to the family, the search time is also dramatically reduced when it comes to identifying the seed. Can we detect the family? It is possible, yes, because in many families, although the species produce seeds with their own different characteristics, some families have seeds that have similar characteristics, a sort of kinship, and this allows us to recognize the family. This is the case of the Asteraceae seeds. Regarding the dispersal units, they tend to be one-seeded fruits, and they disperse on their own or with another structure. Feature and characteristics that make them similar. These are usually elongated seeds. They can be straight or they can be curved. The apex or both ends are truncated, and many times we can see longitudinal grooves on their surface. On the ends, we can find, for instance, on the apex, on the apical end, we find the pappus, which is a structure that characterizes Asteraceae, which is formed by a number of elements that can be morphologically different interspecies. They can be sorts of filaments or scales. They might look different, but they are constant in the different species. Pappus can remain after maturation, after maturity, so we can probably see it in the lab. Or they might easily fall, as is the case in of this seed on the right. When the seed matures, the pappus falls and it doesn't remain. In other species, we will find that the most apical section of the seed has a sort of rim here at the top that is different from the rest of the seed. And at the base, we find a thickened tissue that is also different from the rest of the seed. The Fobasier family also has seeds that have their own characteristics. This allows analysts to recognize seeds as belonging to the family. One of the general and typical characteristics of this grasses family is that the seed wall and the fruit wall are merged, and this creates a specific type of fruit called Carryopsy, which is typical of grasses. This Carryopsy can actually propagate on its own or with other structures. Morphology of this Carryopsy. Most of the Carryopsy surface is covered by the endosperm, and the embryo is located at the base of one of the sides of this Carryopsy. It can be seen externally because there is a change in the surface or in the color or texture of the surface, as we can see in the image. If we look at the Carryopsy from the other side, we will find a sort of scar. It can be a longitudinal groove, more or less deep depending on the species, or it can be a circular groove as well. When Carryopsy develop, they are included within a structure formed by two bractea, which are morphologically different. These are the lemma and the pallia. The set of this lemma and pallia actually make up what is called the florets. Typically, these florets appear along an axis, a Requila, in an alternated way. At the basis of that Requila, next to the base of the first floret, we have two different elements called the glume. And this is how a typical grass spikelet is formed. At the basis, we have the glume, usually two, and towards the top, a number of florets. They are fruitful when they have a Carryopsy or potentially have a Carryopsy inside. There are variations regarding this typical spikelet structure, especially the number of fruitful florets. And in many species, there might be spikelets that just have one fruitful floret and maybe a sterile floret. Regarding dispersal units that we might find in this family, we might find free Carryopsy. If this is the case, it's very easy to identify them because of their typical morphology. It's easy to detect them as part of the grasses family, but it's very difficult, even impossible, to identify the species or to identify the genus. Many dispersal units are actually florets, and there are species where the dispersal takes place with a full spikelets, so we find the glume and a sterile floret, for instance. And there are other dispersal units, the spikelets and other accessory structures. Have a look at the right bottom image. The dispersal unit here does not look like the grasses family, but when we look inside the seed, we will find a Carryopsy with its typical morphology, and this will help us detect the family. Although a free Carryopsy is difficult to identify, if we have more structures in the seed we are observing, then we will have more information to actually help identify the seed. This is something that it's a doubt many people have when it comes to differences between Festaca arundinacea and Lolium multiflorum. In both cases, the seed corresponds to the floret, and the floret is made up of the same elements. So what information do these elements provide us with? In the case of Festaca arundinacea, these are the pictures that have an A, and Lolium multiflorum, they have a B. Species A, and the typical shape is elliptical. They have a central area of the seed where we find its maximum width. However, typically species B is of an oblong shape. Therefore, its margins remain parallel through most of the seed's structure. In both cases, we have a callus, a thickened tissue. And have a look at the images on the left. Here we have the seeds with the lemma seen from the front. Have a look at the base here in this position. Between the lemma's base and the callus, there is a depression, a wide depression in species B, and that depression is just a tiny groove in species B. Another element that helps us identify these seeds is the Requila that is shown in the two images in the middle of the slide. The Requila is a cylinder in species A, and its apical area is disc-like in shape. It tends to actually separate from the seed. In species B, the Requila is flat, and it remains on actually the seed. There are also differences regarding the palea. In both species, if we observe the bottom, in both cases, we need to analyze it. We need to observe it with a stereoscopic microscope. We need further magnification, but we can see here the different spots. And in species B, those spots continue up to the top, but in species B, the spots are lost and the pallium becomes brighter. The third family in importance is this Fobasier family, and within them, the subfamily Faboideae. It is the most important one because it has a highest number of species. This subfamily has around 13,800 species. Seed characteristics. They vary, but there is one characteristic that is quite important. And when we see it, we know that this is a a seed from this subfamily. And it's this hilum that we can see, this sort of scar that we can see on the surface. It can have different shapes or size depending on the species, but there is always this longitudinal fissure that included throughout the hilum, and this actually ensures that this is this subfamily. There are other families that have their own characteristics, Apiaceae, Brassicaceae, Malvaceae, etc. There are more, of course. An analyst should know that devoting time to studying these features actually increases the chances of successfully identifying the seeds and doing this quickly as well. Thank you very much. I would like to thank you for your attention. Thank you, Anna. Your presentation has been very clear. Of course, you have talked about analyst training and analyst ethics as well when it comes to conducting the analysis. Now, I would like to read out some questions received from Mexico. Juan Manuel Chavez. Within the concept of seed, what do you consider this propagation material, seedlings, and other types? Well, actually, the popular term seed includes all the vegetation multiplication organs that can be used to breed the species, for the species to breed. And it doesn't necessarily need to be look like a seed, but it should actually fulfill this role of breeding a new generation. In this sense, we can call them a seed. For instance, the potato is a seed. When it comes to these organ multiplication organs. Another question, can we detect different low-limb varieties through a physical analysis and a diploid from a tetraploid species? Varieties, sorry. Well, low-limb varieties, no, it can't be done. Different low-limb cultivars, no, this can't be done. There are many low-limb species which are very difficult to differentiate. Therefore, I would say that that is not possible. And the next question was, is it possible to distinguish a diploid from a tetraploid variety? Although it's normal for tetraploid varieties to have larger seeds, there will always be large seeds, but as the different florets that are part of a spikelet are not of the same size, there will be a variation in size once again. Therefore, smaller florets within a tetraploid spikelet may be similar or the same as the larger seeds of a diploid. So this distinction is not very easy because information is not accurate. Therefore, I would say that this can't be done.
So we outsource the service to institutions that have the proper instruments or tools to do this differentiation and come to an accurate number. And now they are asking about the differentiation of species. They ask, is it possible to differentiate a Lolium multiflorum, or perennial with a physical analysis? No, I would say it's not. Even in the case of Lolium genus, where we have to identify the different types of Lolium to see if they there are some parts they meet they have the same characteristics, but to do this we use the fluorescence technique to be able to differentiate them, to see how much contamination there is in a perennial or the other type of Lolium. I was about to thank you, but I have another question. And this would be the last one, and then the next person will come. How much time do you think an analyst need to have the tests ready according to the specific standards? I would say this changes from case to case, but as a minimum, it should take 1 year because of the species being seasonal and the weeds that come together with them. And it's also important the amount of species that we work in the lab. So how many species we are able to test. But I would think that at least it should be 1 year between one test and the other. And the training to recognize, to see this, to get to know about this big challenge of identifying unknown seeds, it could take a little longer even. There are questions in the chat, so I would like to invite you to ask questions directly to Anna. You can write to her so that we don't delay the rest of the presentations. Thank you very much. She was handling something that has a lot to do with the analyst training, and that the analyst has to take into account when carrying out the tests. It has the advantage of having clear procedures, clear instructions that help the analyst to do the work in a proper way. I would like to introduce you to, Victoria Valtieria. She has a BA in sciences. She is a professor and researcher in the Faculty of Agronomy in the University of the Republic. Good morning to all of you. Thank you, Vanessa, for your introduction. And thank you for your presentation. And I will talk about the names of plants and why it is important to know the scientific names. So the content of what I am going to present is as follows. I will introduce the idea of taxonomy. I will present the background of modern taxonomy. I will present the main taxonomic categories, how it's organized, then the way and the scheme to give a scientific name to species, which are the criteria to build the scientific names. And then I will propose to see the importance of scientific names and why they are important in comparison to common names. And then I will give you some guidelines about databases that are local, regional, or international that can help you identify the current status of the names and then get more information. As I was referring, I will introduce the concept of classification. So taxonomy is a science in charge of classifying, and the purpose is to organize the bodies in a hierarchy. So there are two main activities, nomenclature and identification. So through nomenclature, what we do is define the principles and the rules to give a name to the bodies. And identification is the activity carried out when we have a body in front of us, we don't know the identity, and we put it in a classification that already exists. So these are the basis of the classifications, the basics of the classification. And besides organizing the different bodies and put them in some hierarchical organizations, it aims at showing the kinship relations between different bodies, and we recognize that as phylogenetical trees. This is the scheme that we see in this on the screen, is one of these trees. that is showing the different parts of this tree. And the three main groups that are shown, the relations between these groups is this plants that you see in the different slides, the plants with flowers, the pine trees, and the other ones. So what we can see here is gymnosperms and angiosperms. They are more related than with the one on the left. This is a feature that they share the presence of seeds, the two on the right, and the one on the left has these spores. So in a natural classification, besides the hierarchies in the organizations, we show the evolution, the historical evolution of the different plants. So that's one of the purposes of the classification, not only to organize them, to have them in an easier way. We show the historical evolution of the groups, and that's very important for the classification. If we go to the background of modern taxonomy, I wanted to tell you about Carolus Linnaeus, also known as Linnaeus. He was a professor, a physician, studying physics and nature in the 18th century from Sweden, and he is considered the father of modern taxonomy. He was the first one to define the categories in which the biological classifications are organized. And he was also the first one to propose rules and principles to define and name the different bodies in plants, animals, and other living beings. So I will focus on his organization for plants. So his work was entitled, the title of his work was Species Plantarum, that was published in March 1st in 1753. And this work is a summary and presentation of the descriptions and of all species that were known up to that date. And what I wanted to highlight to that point was that the date it was published, May 1st, 1753, is a date that is set as the start point of a botanical modern nomenclature. It's the beginning of the regulation of scientific numbers with botanical nomenclature. So here what we can see is one of the pages, an example of a page of the book Species Plantarum. And I will tell you in more detail each of the parts, but it's important that something that Linnaeus was proposing, he was organizing the binomial nomenclature. And I will explain what this means. Up to that point, up to before Species Plantarum, the names were the polynomial names. It was a sentence with the name. So it was the genus name of the group plus a description, like a diagnosis description with a polynomial name. That was the way the species were named. So this is one part of Linnaeus' work. We see that he's showing species of the Brassica genus, which you see in capital letters on top, and then the polynomial name, the diagnostical description of the species. After this information, he was putting all the synonyms and the geographical distribution data. The new thing he is proposing, that Linnaeus proposed with Species Plantarum, that in front of the description of the species, he was putting the vulgar name or normal name. This is the term that was used as a regular or vulgar name. So after this work, then this name was associated to this name, and that's when the binomial nomenclature started. This trivial name that was put by Linné in the beginning of the paragraph of each species, he did it almost in the end of the publication. The publication was already going to be, it was in the edition process, but he had this idea of putting it there to facilitate communication. He was not thinking about creating this binomial nomenclature. This came later with the use of the information. It was so easy to share and to use that it was we started using the binomial nomenclature. If we go to the classification, biological classification, they have a hierarchy that includes big groups, very wide, large groups that include smaller groups. And the groups that are smaller, they are included in the group that is above. So the widest group that includes more, the largest number of beings is the dominium, and the smallest is a species. And that's a basic unit that we use when we want to describe the biodiversity. The basic unit is a species. Each of the levels of this hierarchy is a taxonomic category, and the group of beings that are in each of those categories, each of those levels is a taxon. So I was showing in the beginning the main categories, the ones we use most. As you saw in the former presentations, the family, sometimes the order that is above the family that includes several families, but then we use order, family, genus, and species. But there are some intermediate categories that are also used that are important in the groups that are very diverse, as I was referring, the most diverse families like grasses, and so on. The intermediate categories that are the subfamily and tribe, they are important to organize this information. Just to put this in the context that we have an example of Lolium multiflorum. That's a taxon that is from the category species, that is part of a genus that is Lolium, that is included in a tribe Poea, and it's part of a subfamily Poideae and the family of the Poaceae. So if we put this in pictures, we have Lolium multiflorum, that is the species in the Lolium genus multiflorum, and many other Loliums like Lolium perennial Lolium. And then in the Poea, we have several genus, Lolium festuca and activis. And in the subfamily, we have several tribes like the multiflorum, the Poea bromea, like Bromus cartarcticus or Tipeades, and so on. And then including the families Poaceae, it has several subfamilies, Poideae, so all the different families. And now we will see which is the construction of scientific names. As I was pointing out in the beginning, starting from the work of Linné, the scientific names, they are written in Latin and they are binomial. That was established after the work of Linné. Then what does it mean that it's binomial? That it has two terms. The first term is a genus and the second is a specific name. So the genus is always starting with capital letters, and the specific name, I will tell you how it is formed, but it's all in small letters. So before it used to be, initially it was used to in italic, some years ago we started it's free. You can use italic or not to write the scientific name. In the presentation I was using italic, that is the one used traditionally. Then after the name of the species, you have the name of the author that identified the species, and it is followed by the year when it was published. You will see in some references that you also put the reference to the publication where it was first described. By the scientific name of the species is a binomial, is a genus, and then specific name. You probably have noticed that for one same species, there are several scientific names. According to the different code for each species or taxon, we accept only one name as the right name. So you have the example of Lolium multiflorum, and Lolium multiflorum is the accepted name for this species, but there are different other names that are valid, that are different, that were applied to the same species. They are like synonyms. In this synonym list, how do I select the right name, the accepted name that I have to use? There you do it using the priority principle. You see the name that was validly published, and that is the oldest. So in the case of Lolium multiflorum, the date it was published, the name was published, was 1778. That's the oldest date. So the name that has to be used as the right one is that one. And the right name is only one. The other ones are used as synonyms of the right name. And in the code, we set this rule and we speak about right names. But if you go to the documentation and databases, you will see that they use the accepted name as an alternate name to the right name. So they use the accepted name to define the right name. But in the code we only handle the right name or correct name. In the scientific number, there is a nomenclature name. It's what is associated, permanently associated, since it's published. The name of a species is published. This is a sample that represents a species that has been published and saved in a referential herbarium, and it was referred, where it was stored and saved in the original publications. There are different types of nomenclatures, but what is important is to know that for each name, it has one example that is associated. As I was saying before, we have only one correct name and several names that are synonyms of this name. We have different synonyms. We have the nomenclature synonyms that are different names applied to the same species that were described, starting from the same herbal example. And the taxonomic synonyms, these are different names applied to the same entity, and they were created from herbarium material, different material. So how do we build the scientific name of a species? It's a binomial. The first is genus, and then is a specific name. It always has to have an agreement in the genus and the specific name. It has to be agree grammatically and in the gender, in between the genus and the specific name. And there might be different ways of designing the specific name. It can be a noun, nouns that are Latinized, and They are made in the honor of some, for instance, Rosenguti is referring to Rosengut, Bernardo Rosengut, that was an agronomist and grasses studier that was doing it. Other ways of designing these names is with adjectives.
Therefore, they show a specific characteristic of the species. This epithet shows, for instance, that the lolium species has several flowers. An epithet can also be designed by using a geographical adjective that denotes its geographic distribution or some sort of a habitat feature or anything that has to do with the ecosystem where the species exists. In the case of Po uruguayensis, this expresses that it's a species that typically develops in Uruguay. Also, this is the meaning of certain abbreviations. For instance, if we have a lot and we need to identify the seeds, we can identify the genus. For instance, we can say that there are old Hodium species, but we don't know exactly what the species is. So we write SP because we know that it's a species within the genus or Hodium, but I cannot quite identify the species. And the abbreviation SPP is used to express that I cannot identify the species within the sorghum genus. I have the sample to identify. I know that there are different sorghum species. I know the genus, but I cannot really identify the species within the genus sorghum. SPP means that there are several species within the genus sorghum. Regarding common names, I will give you two examples. First one in a herbal species, another one in a wood species. And why are common names a problem? Because sometimes different common names can be used. Maybe a common name is used somewhere in order to identify species, and in another country they use that common name to identify another species. So common names vary worldwide. There might be several common names denoting the different species, and sometimes one common name can be used to identify different species. For instance, in Uruguay we have a common name which is Anacahuita, used to identify Anacardiaceae family, or one species within that, which is Scinus molle. This name, Anacahuita, in Argentina is used to name a species which is a Blephalocaris salicifolis, as you can see in the image. So these are two different species, and Argentina uses the same common name. So these common names do not provide us with any accurate information. Regarding the herbaceous species, I wanted to show you this example of the foxtail grass. There are several species in Uruguay where the common name is used, but one of them belongs to the Cordillera genus. If we analyze the whole of the River Plate area, foxtail grass is a name used for over 25 species within the Poaceae species, and also other species as well. For instance, the species called foxtail grass within the Asteraceae family, Solidago chilensis, also has other common names. So I wanted to show this to you because it's very interesting. This foxtail grass name is used to identify, or used not to identify, over 30 species located in 6 different families. This is a phylogenetic tree of different vascular plants, And I wanted to show you here in bold type the location of these 5 families where we can find this species commonly known as foxtail grass. So using this name, cola de zorro in Spanish, does not really help us regarding its characteristics, its family, and its position within the general plant classification. This is why it's always important to use the scientific name of species, which is the only one that can provide us with real information. To finish, I would like to say that all the principles and rules defined to regulate the use of names is done through the International Code of Nomenclature, which is a publication that is available online. And it is revised every 5 years in a specific session within this Code of Botanic Nomenclature. The latest edition was held in China, 2017, Shenzhen, in the International Botanic Congress, and it was published in 2018. This is also available online. Regarding databases, they allow us to verify the name status. We can also check the spelling of scientific names. Here in this region, we use a very interesting database, which is called the Darwinian Botanics Institute. You can see the link on the slide. All the work done is based on dissertations or monographs. So they have been studied in depth. Internationally speaking, there is a solid database called the one prepared by the Kew Garden in conjunction with herbal institutions from Australia and Harvard. And this database is also a very interesting database, IPNI. A further database that you can use is tropicus.org. It is edited by researchers from the Missouri Botanical Garden. You can, of course, look up the scientific name, the genus, the family. This database includes synonyms. It also shows you the right name. And it includes links so that you can see typical examples, typical samples of the names. Thank you very much. Thank you, Victoria. Of course, this is very important for analyst training as well. And of course, we can make mistakes when we use common names instead of scientific names when identifying seeds. Of course, your presentation was very clear regarding the source of the names and its importance and their importance. Here we have a question. What is the difference between an accepted name and a correct name? Maybe you mentioned this, but maybe it's not clear. Actually, the code mentions the correct name. For a taxon at the species or genus level, of course, there are some exceptions at the family level, but that is included in the code. The code talks about correct name. There is one name, and it's the correct name. What happens is that other names are used, the word accepted is used instead of correct. But within the code, the word accepted name is not, the term accepted name is not used. But some databases talk about correct name or accepted name without making a distinction. Another question. Why do species names change? Well, we can't help that. In the past, there used to be many researchers that identified species locally, but they, of course, communication was not the same as what we have today. So now, in the history of research, these names were collected or were they described independently. What happens nowadays is that although the biological classification is conducted for names not to vary and to remain stable, in the last few years we have also included molecular analysis to systematic and taxonomic studies. This brings in a lot more information. And as we have so much more information, we need to remember that our aim is to have monophyletic groups that share a common ancestor, as I was saying at the beginning. The classification aims to show the evolution history of the groups. And when there is so much molecular information, those groups become sort of undone. And once the structure changes, many times the names change as well. So in brief, it might happen that a species that I thought belonged to the low-limb genus is now found under a different genus. When that happens, the names need to be changed. The idea is for the system to remain stable, but there might be name changes, and that's inevitable. Thank you. Thank you very much. And if anyone has any further questions, they have your contact information. Now let us talk to Teresita Farras. Teresita is an agronomist from University of the Republic, Uruguay. She is now a private advisor regarding analyst training, and she advises laboratories on maintaining their certification. She worked at INASE Uruguay for many years. Welcome, Teresita. We worked together with Teresita for many years at the laboratory, and now she's here again. Good morning. Today, we will be talking about the physical quality in seeds. What does this mean, and how can we evaluate this? Before going on to the first slide, let us talk about the term quality. Its definition changes. And many times nowadays people talk about process quality, environmental quality, and of course, seed quality. Sometimes when they ask us about why it's good to have quality seeds, we know that it's important to have quality seeds when it comes to sowing, because they are the starting point to have a solid production. Let us now talk about seed quality in broader terms, and within quality, let us talk about the physical quality, which is today's topic. What is the meaning of seed quality and which are its elements? A good quality seed has a number of desirable attributes that allow the crop to set with healthy and vigorous plants, and that represent only one variety. Regarding seeds, quality can be subdivided into 4 basic attributes. They have been mentioned already: genetic, physiological, physical, and health related. But I will address them briefly. Genetic attributes. They govern the genetic makeup of the seed. Physiological attributes are the seed's capacity to germinate and by producing vigorous and uniform seeds. Health attributes entail the presence of pathogens such as fungi, viruses, bacteria, etc., that spread various diseases. And finally, we have the physical attributes. They can be affected by any type of contaminant other than the desirable seed. These contaminants, there can be different types of contaminants, different types of inert material, inert matter, maybe the presence of other species. Within physical attributes, we also find the weight of 1,000 seeds and determining the moisture content. In any of these 4 attributes, weakness introduces a limiting factor. And this means that the crops will not be as productive. I will be talking to you about the physical attribute. For seed quality. A good quality seed has a high percentage of pure seed, a minimum quantity of inert matter, a minimum amount of other species as well. They have a low moisture content, and their size and weight represents the whole variety. There is also uniformity regarding size and color. How can we evaluate this physical quality? There are different analyses. Different analyses can be conducted to determine the seed's quality. For instance, purity analysis, determination of other seeds in number, the weight of 1,000 seeds, and determining the moisture content. These 4 tests can be found within the ISTA rules, and each of them have a specific chapter. Of these 4 tests, we will be focusing on the first ones: purity analysis and determining other seeds in number. The other 2 are important, of course, but in the first 2 tests, as you have already seen when Fabian talked about specific standards, these are the 2 tests that form the basis of all our work, and this is why I will be focusing on them today.
So we will start with the purity analysis. The purpose of this analysis is to determine the different components that are part of the seed lot to be tested. It is important in this case to see which are the components in this purity analysis: the pure seed, inert matter, and other seeds. Among other seeds, we have crops and also weeds that are the common weeds and the noxious weed. Let me see here. So the pure seed is a species that is that the one who submitted the sample says he was sending, or the one that is the majority in the test. We have to consider all the varieties that are part of the species. The analyst must take that into account. In the purity analysis, we don't do the avarietal identification. For the case of willing to determine the mix of varieties, then we have a different type of analysis that we will see later. The separation of seed is done through the visible characteristics of the seed that has to be taken into account. And we have to ask ourselves the questions, what is a pure seed for the species we are analyzing? And to determine what is considered pure seed in each species, we have to go and check the definition chart of pure seed from ISTA. Here we have the chart that is a 3B part 1 chart. That is a number of definitions of pure seed and chaffiness of seed listed by gender. It has 4 columns, genus, family, the number, and the chaffiness. In the case that is not our case, if there was chaffiness, you would have a C letter in the 4th column. Here we have 2 examples that we will continue using in our work, so we can have it very clearly, so that you can have an idea of how we carry out a purity analysis and we determine the presence of other seeds. We chose Glycine and Triticum from Fobasier and Fobasier. So, and at the same time, you can see that they have a number 11 for the first one and number 40 for the second one. This number is defining the pure seed, in this case, for the glycine, and the one for Triticum in the second one. Well, if we continue with the same example, we have that they consider pure seed the seeds that are in one piece. That is, the seeds that are in a homogeneous size and shape. So also pure seeds are immature seeds with some damage, wrinkled, germinated, sick, unless they are partially or totally transformed in sclerosis and in galls or smut. So we find in a purity analysis, here you have all these possibilities, and we will see the ripe seed that are usually in a green color, that are not ripe, sorry. This could be because of 2 reasons. The first, or mainly because of environmental factors, or that it was damaged by insects or some disease. The second is a soybean seed that has some damage. It has a cut. It's a mechanical cut that takes place when it's harvested, and it usually happens when the seed has a low moisture content. Then we have this one and the one at the side where the tegument has some damage. One is cracked and the other one has some wrinkles. Any of them, of these last 2, of these 2 problems, these are environmental factors that are not favorable. So this could be in the first one that is cracked. It could be because of the temperature changes during day and night, and the wrinkled ones, it could have to do with hydration and dehydration processes. We have seeds with a bud shape. This is a genetical component of the seed. Then we have some seeds that are damaged by insects. Sorry. I cannot point at the seed, but you can see the damage. Then you have seeds with a mycelium, with a fungi. The seed is carrying it, and it could stay for a long period of time. Here we have seeds that has some purple spots that is caused by this Cercophora ticuci. It could be a small area, or it could cover the whole seed. And finally, we have this case that has some spot that is caused by a disease. It's kind of brownish or black that starts in the hilum of the seed, and it goes, and it might even cover the whole seed. Same with the wheat seeds. We have seeds that are green or greenish. We have seeds that already started germinating. Then seeds where the embryo is eaten by larvae. And the last two on the bottom of the slide, we find seeds with holes that are made by insects, the first one, and some seeds that were attacked by smut by fungi. In the case of the two on the bottom, the analyst must take into account that despite the holes or the damage caused by the fungus, the healthy part of the seed is, if the healthy part of the seed is bigger than the one with damage, and that could be considered a pure seed. So we have to identify if it was not losing the shape totally, so it could be identified as wheat seed anyway. Other things that are considered pure seed are parts of seed that are bigger than half the original size. Taking into account that one part of the testa is still there, and the cotyledon is broken, but is still attached to it. Then pieces of seed that are bigger than 50% of the original size. The second component, as you might remember, of this purity analysis is the inert matter that includes seed units and all the other materials and structures that are not defined as pure seed or other seeds. There are seeds, broken seeds or damaged that are equal or smaller than 50% of the original size. Here we, on the case of wheat, we have the one to the left is a pure seed, and the other two pieces are inert matter because they are the half the size or lower, smaller than half the size. When we find halves, we have to, in a purity analysis, we have to compare it with the size of the seed we are analyzing or testing at that moment. In the case of Fobasier, we have separate cotyledons, and then we have seeds and parts of seeds totally separated. So if they don't have this, it's not attached to everything, then it's inert matter. All other matters that are materials that are not seeds, parts of tissues, vegetable, leftovers, soil, insects, parts, and some parts of fungus, that are hard, that they are like that because environment conditions are not favorable, and they are just waiting for favorable conditions to develop. Another component, and in fact the last one in the purity analysis, are other seeds, and it involves some units, dispersion units of any other species, crops or weeds that are not the pure seed, to comply with the specific standards, quality standards in Uruguay. This is separated in seeds of other crops from weed seeds. Among the weed seeds, we have the noxious ones and the other ones, and we have the ones with zero tolerance for other species and with some tolerance according to the species that is being analyzed. Now we will move to the determinations of other seeds in number. That was the second of these steps to assess the physical quality of the seed. And the purpose of this is to establish the amount of seeds of other species that were sent by the one who submitted the sample, like estimate the number of all of the other species, the number of a special category of seeds like noxious species for a special country, and the specific number for a specific species. So the scope of the determination of other seeds will be informed as a complete analysis, limited, reduced, and limited, reduced. The complete analysis, we will analyze the whole weight of the work sample, identifying all the other seeds that are present. In the case we were analyzing that we were working, soybean and wheat, what the complete analysis is telling us that we analyze 1 kilo of wheat or 1 kilo of soybean. And when we analyze them, we identify all the other seeds that are present. In a limited analysis, we analyze the whole weight of the work sample, but for one or more species sent by the one who submitted it, or to comply with national or international standards. We still work with the total weight of the sample, but we won't separate all the seeds that are found, but only the species that are pointed out by the different standards. Our specific standards are based on limited analysis. As you saw when Fabian was making his presentation, in the analysis we will look for seeds from other crops and the tolerated with seeds for the ones, the species, the concrete species. The reduced analysis, we analyze a smaller weight of the work sample, identifying all the other seeds that are present. This is usually used for small lot seeds. The reduced limited analysis, we analyze a smaller weight of the work sample, but for one or more species that were sent by the submitter or to comply with national or international standards. It's also used for species that are difficult to identify. And in this case, we analyze 1/5, a minimum of 1/5 of the sample. This can be used as an example when we are analyzing, or our work sample is some special type of grass, and we have to identify sorghum. So we use 1/5 of the sample that is determined by the chart. The determination is done by through counting. It's a visual identification, same as the purity analysis, to count the structures that are found as seeds. It will be taken into account the definitions of pure seed. This is important because sometimes we find fragments, parts of seeds or other types of seeds, that if we go to the identification of pure seed, they are not considered pure seed. And so we shouldn't take it into account when we are counting. And then we have seeds that are grouped in fruits, or so they should be counted individually. This happens in Antimyxcotula. We have to separate each of them and count them. And the same if it's like the fruit of Raphanus sativus, we break it and we see how many seeds it has inside. It can happen in other species because there might be pure seeds and it can happen that we find seeds that are not considered pure seeds. So we have to separate that. Well, this is the end of my presentation. Thank you very much. They are telling in the chat that your presentation was very clear. They keep your contact for any question they want to make. So, and you were able to go straight to the point. So this was really great. You were able to bring us to the analysis, the purity analysis. We have a question. If we have to make notes and take it into account when germinating, when to want to choose a substrate, if we have to take into account fungi or insects presence. It is important when we analyze purity, as we saw in the case of wheat or in the case of the soybean seed. We saw that wheat seed that doesn't have the embryo, that won't germinate, but we have to consider it as a pure seed. And same with the soybean seed that is cut, mechanically cut, or it has been bitten by an insect. If it's been bitten by an insect, it will depend on the area and how deep it is. So if we see we have a wheat seed that is bigger than 50% of the seed, and it has, so it's very clear, the standard. If we remove all those seeds and we don't take it to the germination analysis, we will our evaluation of the lot will be wrong, because it will be better than reality. So how for each seed do you find the categories of the tolerated, non-tolerated, and so on, accepted? We are analyzing a wheat seed. We will have to see in the lot, we go to the standard and we see which is the maximum number of presence of other crops to comply with the standards, and same with the weeds. If we find forbidden weeds and the noxious weeds, the common ones, we leave them aside. It's very clear. There are questions that were not answered that are in the chat. We are very thankful to you, and we close the presentation cycle now.
What should be done if an analyst or laboratory cannot identify a seed in a purity test? Fabian, you have the floor. Well, first of all, they should check with other analysts at the laboratory who maybe are more experienced, so that they can work with other specialized laboratories or other companies that specialize in seed identification. Anna, what do you think? Yes, I believe that they should make the best efforts to identify the seed. They should also remember that if they cannot identify the species, they can report the genus and the family. The rules clearly state that we can report the most accurate taxon as possible. I fully agree with Fabian that they can even send the sample to the university or to INASE in Uruguay as well, so that we can help you identify the seeds. So you should do they should do their best in order to classify the seeds. And they might even report that they have found a seed that they cannot identify. As Anna was saying, and as she also said during her presentation, I believe that having the family already is positive. Genus, that's great. And as Fabian and Anna was saying, there are other analysts and colleagues they can resort to, and also botanists. Maybe they have some more information besides the fruit or the seed. And I'm sure that a taxonomist or botanist can also help identify the seed. Thank you, Teresita. I think everything has been said. There are also websites nowadays where you can try and find the seed. Laboratories should have a solid seed collection, and they should also be in touch with other botanical gardens, for instance, and they can also resort to the botanics department at the university, which we have worked with many times, to help us identify the seed. In that case, for instance, if we're talking about an export situation, we need to comply with the country's standards. We need to identify the noxious weeds, and we can sometimes, when we write identify in an analysis certificate, then the receiving country might have a problem if we write without identification. So we might, for instance, we might write that this is not a weed or a forbidden weed in that country. Also, Teresita was talking about websites, and there are many websites with great pictures that help us identify species. We must remember that many times we see other regions' flora, and there might be some species with similar seeds. And if the image is the closest to the seed we found, we might tend to believe that it's the same species. But maybe that lot was produced in Uruguay, and there's a species that is similar to the one in the picture, same genus, for instance, but it's a different species. So if possible, please check the source of the seed lot. Verify the species to check that the species appears in our region, because maybe we're reporting a seed that doesn't feature in our region in Uruguay, and we are saying it is. So that is something to be considered. Earlier we talked about the requirements that analysis labs need to fulfill, and also we talked about the seed collection. How does a laboratory expand the seed collection. Maybe there's a lab that would like to apply for authorization. Maybe they have a seed collection that they haven't been able to enlarge. Victoria, what would you say to this? Although I am not a specialist and don't work with seed collections, I think this should be as in a herbal institution, a herb repository. Also, they could exchange seeds with other seed banks, as is the case of herb repositories. They could swap seeds, for instance, and they could swap samples, and in that way they can enlarge their collections. Great, thank you. Teresita. Besides that, when we have authorized labs and we send them reference tests, and this is done at INASE to see how the lab is working. Of course, the work sample is contaminated with different weeds and seeds and other seeds. Then they send us the data and we tell them what the seed was. We always tell them to keep the seed if they haven't identified it, because it's a way to enlarge the seed collection. And they can also get in touch with other labs. For instance, a laboratory has many seeds and processing plants as well, where we find several weeds in order to conduct these referee tests. Well, I agree with Victoria. It's great to exchange seeds with different organizations, even from other countries, as is the case with INASE. And this is beneficial. This benefits both institutions. We can swap seeds with this institution. When there is this exchange between laboratories, the seeds need to be correctly identified, because it's important not to, let's say, misidentify a seed and then perpetuate the mistake. The seed needs to have been checked by the university or an identification institution, etc. Thank you, Fabian. Anna. Well, I can't think of any other way. Usually when an analyst is just beginning the seed collection, they will put it together with the seeds they work with and find at the lab with the help of more experienced analysts. To enlarge the collection, you can even buy different seeds at greenhouses. If you can go out into the field and collect plants with seeds, you can ask the university to identify the plants. And there you will have this accurate identification, because the plant provides a lot more information than just an isolated seed. So the best way to, this is the best way to increase the seed collection. And the rest is applicable as well. Thank you very much. I believe that this analyst will find all your replies very useful when it comes to enlarging their seed collection. Thank you very much to the four of you for your time, devotion, and the quality of your presentations. And please stay with us for the rest of the meeting. Thank you.